students, I welcome you all for our lecture series on agrochemicals and pest control. The topic for today's discussion is pests of pulses and sugarcane. We all know that pulses play a vital role in our lives. Pulses are the cheapest source of dietary proteins. The high content of protein in pulses makes the diet more nutritive for a vegetarian when taken with other cooked food items. The pulses are also known for increasing productivity of soil through fixation of nitrogen from atmosphere, addition of biomass to soil and secretion of growth promoting substances. Pulses are well suited in rain-fed conditions and require less farm resources. Hence, farmers prefer to grow them from economic point of view throughout the country. Pulses are grown globally, covering large dimensions of about 70.50 million hectares in area with a total production of 57.27 million tons. Amongst different pulse producing countries, India ranks first, having 29.96% of the total pulse acreage, though it contributes only 22.52% of the global pulse production. And so, in this episode, the following aspects are going to be seen: pests of pulses, eriophid mite, aphids, thrips, major insect pest and management of sugarcane. In the first part, we are discussing about pulses pests, namely pod borer and pigeon pea pod fly. Insect numbers can build up quickly in pulse crops and, if left uncontrolled, will substantially reduce both the yield and quality of the grain. It is important to monitor insect numbers carefully because the large size and high value of most pulse grain makes crops vulnerable to relatively low numbers of insects. Control strategies must be applied as soon as problems become apparent. Crop establishment to flowering through podding are the two key stages of crop development where insects are likely to be a problem and when careful monitoring is required. Sweeping crops with an insect net is the best way to monitor most insects after the crop is established. Pod borer. The scientific name of this pest is Helicoverpa armigera. Helicoverpa is one of the major polyphagous pests distributed throughout the country. The adult moths have yellowish to orange forewings in females and greenish grey in males, with a lightly darker transverse band in the distal third. The kidney shaped marking is slightly distinct and smoky. Hindwings are pale grey with a darker broad marginal band having small brown marking at base. Adult moths have a wingspan of 35 to 40 mm. Females lay several hundred eggs on all parts of plant but have preference for young shoots and florets. The eggs hatch in less than 3 days at optimum temperature of 27 to 28 degrees centigrade. On hatching, the larvae feeds for a short time on the tender leaflets by scraping green tissue and then shift to flower buds and tender shoots. Larval and pupal periods last for 17 to 23 and 9 to 11 days respectively in normal conditions. The color of the larvae varies from yellow to green, pink, orange, brown or black, but all have characteristic light and dark stripes along each side. Slowly it enters and feeds on the seeds inside the pods. The half portion of larvae remains inside pod while feeding on the developing seeds. They can cut hole on one to another locule and feed 20 to 25 pods in its lifetime. Coming to management of Helicoverpa. Several insecticides including endosulfone, monocrotophos and synthetic pyrethroids give good control, particularly if applied soon after the eggs hatch. However, considerable resistance to many insecticides has been reported in these insects in areas where crops have been sprayed intensively. Helicoverpa species have many parasites and predators. In some areas, these give adequate control and pesticide use may lead to an increase in Helicoverpa populations through the reduction of these natural enemies. Different components of integrated pest management recommended for pigeon pea and frequency of adoption of these practices is depicted separately at the end. The next pest is pigeon pea pod fly. The scientific name is Melanogramoisa obtusa. It is widely distributed in India and causes major economic losses in northern parts, causing significant losses especially in long duration varieties. This pest alone accounts for 70 to 80% of the total pod damage by pod borer complex. The infected seeds do not germinate. There are no obvious external symptoms of pod fly attack till the fully grown larvae chew holes in the pod walls, leaving a window through which the flies emerge after pupation in the pod. Coming to management, as development of all the stages of the pest takes place inside the pod, according to the recent studies, even systemic insecticides such as dimethoate and monocrotophos do not reach the target site to kill the maggots which are feeding inside the pod. Secondly, it is highly impossible to make the whole pod into lethal dose so as to kill the maggots. It is also not advisable for the residue point of view. Once the egg laying is over, then it is very difficult to manage this pest with any of the available insecticides. But non-systemic insecticides like endosulfone, methomyl, acephate help in killing the adults. In this direction, 1% jaggery can be mixed along with the contact insecticides to attract and kill the adult population. A cold season during which no pigeon pea pods are available will reduce the infestation. Late sown and late maturing varieties are vulnerable to the pod fly attack. It is best to avoid growing a mixture of cultivars of differing duration in an area because this will make the pods available over a long period and allow several generations to develop. Some pigeon pea genotypes are much more susceptible to egg laying than others, so it may be possible to select resistant cultivars. The next pest that we are going to study is eriophid mite. Eriophid mite scientific name is Azaria kajani. It is a common and widely distributed insect. It is uniformly distributed on the leaves with major concentration in the younger leaves. More than 90% of the Azaria kajani occur on the lower surface of the leaves in the diseased plants. It is the vector of the pigeon pea sterility mosaic virus causing the most damaging sterility mosaic disease of this crop in India. As a result of the transmission of the sterility mosaic virus, infected plants develop light green or chlorotic leaves which have mosaic patterns. Most infected plants do not bear flowers, hence the name of the disease is sterility mosaic disease. Vegetative buds harbor significantly larger population of mites than leaves. This mite is highly host specific and is largely confined to pigeon pea and its wild varieties viz. Cajanus scarbioides and Cajanus cajanifolius growing as wild plants in the fields. These mites are small, pink in color, spindle shaped and normally feed on the underside of leaflets. The mites are difficult to see with the naked eye. Adults measure 200 to 250 micrometer and have a life cycle of about 2 weeks comprising egg and 2 nymphal stages. It lays milky white eggs on the vegetative terminals. Many of nymphs are found on young folded leaflets. The wind dispersal of infective mites may cause plant to plant infestation and its spread to adjoining crops. Coming to management, it is best to prevent this mite and the sterility mosaic disease by the use of resistant pigeon pea varieties. Several insecticides including dimethoate and acaricides such as dicofol 18.5 EC at 2.5 ml per liter help to control these mites effectively. The third important pest is aphids. The scientific name of aphids is Aphis crassivora. Several species of aphids have been reported from pigeon pea, among which Aphis crassivora is the most prevalent as it is having a broad host range. Aphids colonize the young shoots, flowers and pods and draw sap from them. The immature are lightly dusted with wax and are lightly brownish. However, adults are black and shiny, up to 2 mm long and some are winged. Nymphs are smaller and light reddish brown. Abdomen is black, pigmentation often extending laterally to enclose lateral sclerites and to encircle bases of siphonculi. Antenna is about 2/3 as long as the body. The species can reproduce without mating in creating one generation in a week under optimum conditions. Individually, adults can produce about 100 nymphs over a lifespan of up to 30 days. Aphids seldom become economic pests, hence application of chemical pesticides may be forbidden in favor of naturally occurring ladybird beetle and chrysoperla bugs. Coming to the management, aphid colonies on pigeon pea rarely thrive for long, probably because of the natural enemy activity. Rain also results in a large reduction in infestation. Several systemic insecticides including dimethoate 30 EC at 1.7 ml per liter adequately control aphids and can be used if needed. Coming to the next part, that is thrips. The scientific name of thrips is Megalurothrips usistatus. Several genera and species of thrips have been recorded on pigeon pea. In India, the most common species appears to be Megalurothrips usistatus, which feeds on flowers. In Eastern Africa, other megalurothrips species are common on pigeon pea and are considered important. Heavy infestation of thrips can lead to shedding of buds and flowers. The black adults and nymphs are easily seen with naked eye, particularly when they are on yellow flower petals. One generation can be completed within 3 weeks. Coming to management, in India, the population buildup of thrips on pigeon pea is generally not large enough to cause substantial damage and do not therefore merit specific control measures. In most cases, insecticides such as endosulfone 35 EC at 2 ml per liter or monocrotophos 36 SL at 1 ml per liter or dimethoate 30 EC at 1.7 ml per liter used to control major pests also reduce thrips populations effectively. Coming to the last part in this session is major insect pest and management of sugarcane. Sugarcane is a long duration crop of 10 to 18 months and therefore is liable to be attacked by a number of insects, pests and diseases. According to an estimate, sugarcane production declines by 20 and 19% by insect pests and diseases respectively. To increase the crop productivity, management of insect pest and disease is of great significance. Due to diversity in agroecological conditions, the importance of insect pests and disease varies and therefore management strategy should be adopted accordingly. Sugarcane is infested by about 288 insects, of which nearly 2 dozen causes heavy losses to the quality as well as quantity of the crop. The scenario of insect pests and diseases varies in subtropical and tropical belt of sugarcane. Top borer and stalk borer are found predominantly in subtropical areas where internodes borer and yearly shoot borer are among disease rust and eye spot are prevalent in tropical region. Several management strategies have been developed as a result of research and development work. In order to save environment from chemical pollution, use of biocontrol has been given utmost attention. The management technologies have been integrated as per need for increasing the efficiency. The first pest is early shoot borer. Scientific name is Chiloinfest catalyst. It attacks the crop during the early part of cane growth before internodes formation. It also attacks the cane stalks in the years of scanty rainfall. Larvae enter the cane laterally through one or more holes in the stalk and bores downwards as well as upward, killing the growing point. Thus, it cuts off the central leaf spindle, which eventually dries, forming a dead heart. The dead heart can be easily pulled out. It emits an offensive odor. Borer infestation during the germination phase kills the mother shoots, resulting in the drying up of the entire clumps. This leads to gaps in the fields. It causes heavy yield losses as it affects the plant stand or unit area. It also leads to canes of different age, which will be poor in juice quality with less cane weight. When borer infects cane stalks, both yield and quality are reduced. Coming to the next one, internode borer. The scientific name is Chylosaccharophagous indicus. It damages the crop soon after internode formation and its activity continues till harvest. Lodging, high dosage of nitrogen, waterlogged condition and presence of water shoots favor buildup of pest. Fresh borer attack is mostly found in the top 5 immature internodes. Caterpillars bore at the nodal region and enter the stem and tunnel upwards in a characteristic spiral fashion. Entrance hole is usually plugged with excreta. Larvae feed and multiply in water shoots. One larvae found in a single cane damages 1 to 3 internodes. The length and girth of the infected internodes get reduced. Yield loss and juice quality deterioration occurs when the infestation is severe. The third pest is top borer and the scientific name is Scarpophaga exarctalis. Waterlogging favors moth attack. Larva first tunnels into the midrib of the leaves and causes a white streak which later turns reddish brown, usually in the second to fifth leaf from the top. As a result of biting across the spindle, a number of shoot holes are formed in the leaf. As larva nibbles into the central core of the cane, a portion of the internal tissue is eaten, resulting in dead heart formation. Dead heart, when formed, is reddish brown, appears charred and cannot be easily pulled out. In tillering phase of the crop, the attacked shoots die, side shoots develop producing a bunchy top appearance. In the grand growth period, the crop growth is arrested and the crown with dead heart dries and may be blown off, leaving the stump. Severe yield loss and quality deterioration occurs due to top borer. Depending upon the incidence level, yield loss may be up to 20 to 30%. Scale insect, scientific name Melanopsis glomerata. Waterlogging, high temperature and humidity favor buildup of scale insect population. Rainwater and high wind velocity facilitate dispersal of the pest. It spreads to new areas through seed material. Men and animals passing through the infested fields also lead to spread of the pest to the adjoining areas. Scales usually establish on internodes covered with leaf sheath. The leaves of infested canes show signs of tip drying and unhealthy pale green color and with continued infestation turn yellow. Desapping leads to non-opening of leaves which also turn yellow and finally dry out. Nodal region is more infested than internodal region. Infested crop loses its vigor, cane shrivel, growth is stunted and internodal length is reduced drastically. Ultimately, cane dries up. Such canes when split open appears brownish red. Thus, yield and quality suffers. The yield loss could range from negligible to total crop failure. Now, we can conclude our session. I conclude the session by saying that each pulse crop has its own management practices and cropping pattern. Farmers may not know about all the information, so they need rapid access to all the possible information and take fast decision to manage their crops effectively. In order to have a successful pulse crop and remain competitive, the modern farmers often rely on agricultural specialists to assist them in decision making. Unfortunately, pulse specialists are not always available for consultation at the nick of the time. To solve this problem, an expert system may become a powerful tool and a dire need of the day for farmers, extension workers and government officials.